First, we're going to talk about the absorption spectrum. So as we covered previously in our physics lectures, waves have varying wavelengths, which can change their properties. So light waves have come in a spectrum of wavelengths and as a result frequencies, and this determines what types of light waves they are, whether they're visible light, UV light, infrared microwaves, anything like that. Okay. For example, there's a certain wavelength range which allows us to see the light waves, and that's called the visible light range. That's what you're used to. And within this range, different wavelengths produce different colors. Red light is a different wavelength than blue light, which is different than green light, which is different than purple light, etc. When this light hits an object, part of the light is absorbed by the object, and the rest is emitted from the object. What we see on the object is emitted light. So, for example, if I was to, so you can see that my shirt appears to be blue. What happens is when the white light, which has all of the wavelengths, hits my shirt, the material in it absorbs all of the wavelengths of light, the red, the green, the purple, the orange, the yellow, and the only thing that it doesn't absorb is the blue light. So it reflects the blue light back. And when you look at it, you're seeing the blue light that's being emitted back, right? That's not being absorbed by the material. So it appears blue because that's the only color, the only wavelength of light that's not being absorbed by the object, okay? This happens, of course, in biology as well. And one common example of this that some of you may have seen in a biolab is chlorophyll. You guys know that chlorophyll in plant cells is usually green. It has a green color. And here's a graph that shows the absorbance of different types of chlorophylls and carotenoids at of different wavelengths of light. So you can see that most of the time chlorophylls, they absorb purple, blue, red, and orange light very well, as you can see here, but they have very, very low absorbance of like this green, yellow light, right? So when you look at these, chlorophylls or anything through a microscope or something like that, you'll see that they appear green because that's the color of light that they are not absorbing. So they have a green color or green appearance. So that's all good, and we've learned most of this before. So what does that mean for today's lecture? So the idea of absorption can and is used a lot in experimentation to visualize changes. One common use is in a biology chemistry experiment is to use absorption to track the progress of a reaction. This is a reaction that a lot of you know, photosynthesis, and some of you may have even done this experiment with photosynthesis where you can actually track the rate of photosynthesis in a plant cell. And you can change the factors. So for example, you can see what's the rate of photosynthesis when I put it in outside in the sunlight, what's the rate of photosynthesis when I put it next to like a heat lamp and keep it there, and what's the rate of photosynthesis if I keep it in complete darkness. And you want to see the difference of how like light affects photosynthesis. Well, how you would measure the rate that the reaction is proceeding is through this, at least one of the ways is through absorption. For example, if you put chlorophylls into different tubes and you add an indicator, which in this case is DPIP, but you don't really have to know that. This indicator, it turns from a blue color to a clear color during the process of photosynthesis. The exact way it does this, you don't have to know, but it acts as a substitute for one of the intermediates, and it can accept one of the electrons and get reduced instead. And when it gets reduced, it changes from blue to clear, but you don't have to know that. And so when it does this, you can measure the rate of photosynthesis by simply finding the absorption of blue light over time. So you would put the chlorophylls in a test tube, you would put the indicator in, it would spin around and it makes it blue. Then you would keep it in, say, keep it outside in the sunlight, and then every, say, minute, you would measure what's the absorbance, every minute, and then you can record it, and you can just make a graph of it over time. You can plot it on a graph, and you can find it, and you'll have a slope. You'll have different time points. You'll have a slope, and that slope will be your rate, and that's the rate that photosynthesis is occurring in this specific cell. And so when you have something like this, you can actually like see the way it goes from blue to clear, and the way you do that is through a specific tool you put the test tube in, like I mentioned, you have a test tube, goes from blue to clear. Every minute you take the test tube and you put it in this machine, you set the machine to specific wavelength. So you want to set it to the wavelength for a blue light. It would shine a light through the test tube, and when it goes through, it'll give you like a percentage or like an amount absorbed, and it'll be some number, whatever. And if it's blue, so at the start you expect it to have a very low absorbance because it's absorbing most. It is blue, so that means it's not absorbing blue. If that makes sense. It absorbs all the other colors except blue, so it actually is not blue. And then as it gets more and more clear, it'll be absorbing more and more of the blue light into itself, and that's why it won't be appearing blue anymore, right? And that's just kind of like an example of an experiment. You don't really have to know like the specific experiment or anything like that. It's just maybe one thing that you've done before. Okay, let's talk about the mechanism for this absorption. And the way the absorption is measured is based on electronic orbitals, which we've covered a little bit before, but here is an example of the orbitals. So you have the sigma bonds, the pi bonds. Remember, sigma bonds are like the first bond, and then the pi bonding is the double and triple bonds that's shared. This is the non-bonding, which is where the lone pairs, right? And the anti-bonding, which actually, as it says here, these are normally empty, but these are like to kind of prevent. These are kind of like, as you would think from anti-bonding, these just remove the bonding essentially. Okay, but again, it's not super important. Just remember that the energy levels are important. So it starts off sigma bond, then pi, and then non-bonding, and then anti-bonding, right? I remember it costs more energy to break a sigma bond than does pi bond, so sigma is higher than pi. It's easier to form a sigma bond than a pi bond, so sigma is below pi in terms of energy, right? And the reason that you want to go from non-bonding to bonding, normally from lone pairs to bonding, is because it's lower energy, right? Remember that all light has energy in it based on the formula E equals hv, where h is Planck's constant and v is the frequency of the light, or E equals hc over lambda, which is kind of, I think, in some ways easier to use. Depends what they give you, whether they give you the frequency or they give you the wavelength, because you already know what speed of light is. Where c is the speed of light and lambda is the wavelength. So you can find the energy that all light waves have. And then when this light, which has energy, hits a molecule, it transfers some of the energy to the electrons. The electrons get excited and use that energy to rise to higher energy orbitals, right? So it can go from, as you can see here, from sigma bonding to a pi anti-bonding. It can go from sigma bonding to sigma anti-bonding, right? It can move up. The electrons from lower level orbitals get the energy to move up to higher level orbitals, and that's called excitation because they're higher, they're excited. Okay, the exact amount of the certain wavelength of light absorbed is dependent on what orbitals contain electrons and how much energy they need for excitation, right? Different configurations, bonds require different wavelengths and therefore different energies of light to be excited, and so that would be the wavelength of the highest absorption. I know that was a little bit confusing, but essentially what it's saying is based on how the electrons are assembled for each different molecule in these sigma bonding, pi bonding, and non-bonding orbitals is what plays a role in what wavelength would be best for absorption. Because remember, wavelength is directly tied to energy. So the shorter the wavelength, the higher the energy. If it needs to go up a lot to be, if, for example, the molecule needs a lot of energy to be excited, it would absorb very well a short wavelength, right? It would like to absorb a short wavelength, or might want to absorb a longer wavelength and go to a different level or something like that, right? It's based on what is more favorable based on like the exact amount of energy it needs for exact excitation, right? And that's based on its orbital structure, right? And each molecule usually will have like fairly different orbital structures, right? Okay, so that's the basics of just absorption, right?